Welcome to This Explainer. Today, we're going to demystify the seemingly abstract concept of recombinant DNA technology. If you've ever wondered exactly how scientists manipulate genetic material to create those incredible breakthroughs you hear about, well, it all comes down to exploring the microscopic factories hidden right inside cells. So let's just dive right into Chapter 2 and unlock the secrets of the host vector system. Here is our quick roadmap for today. We're going to cover 1. The gene cloning system. 2. The cellular factory, which are the hosts. 3. DNA delivery are vectors. 4. Essential vector features. And 5. Specialized vectors. Okay, starting off with Section 1, the gene cloning system. So what exactly are we dealing with here? Basically, recombinant DNA, or rDNA for short, is the joining of two different DNA molecules. And the whole goal here is to isolate, propagate, characterize, and manipulate genes for various applications. It's helpful to think of rDNA technology as a two-component system. Imagine a highly efficient delivery truck and a massive manufacturing factory working perfectly together to manipulate genetic blueprints. Getting this job done requires two major steps. Step 1 is isolating the desired DNA molecule from the source. We generally call this the target gene, or sometimes just the insert. Once we have that specific blueprint isolated, we move to step 2, which is actually inserting that target gene into a convenient carrier DNA molecule. That carrier, that's our vector. And together, the vector containing the insert is what makes up our actual recombinant DNA. Now, picture the process like this. First, a process called restriction digestion literally opens up both a target gene and the vector. Then, ligation steps in to glue or join them together. Once successfully joined, this newly formed recombinant DNA is introduced into a host cell through a process called transformation. From there, it basically hijacks the genetic machinery of the host to undergo propagation. This entire continuous cycle, that's what scientists call gene cloning. Moving right along to Section 2, the cellular factory, hosts. A highly effective factory shouldn't just tear up or degrade the foreign DNA the second it arrives, right? It needs to welcome it and supply the necessary enzymes for smooth replication. For prokaryotic hosts, E. coli is absolutely the standout star, specifically the K12 strain. It's a rod-shaped bacterium that grows incredibly fast. It literally doubles its population every 20 minutes. But if we need a eukaryotic host for, say, more complex processes, Baker's yeast, Saccharomyces cerevisiae, is hands down the most widely used complex host out there. Which brings us to Section 3, DNA delivery, the vector. Now, theoretically speaking, any molecule of DNA that can replicate itself inside a host cell can work as a vector for gene cloning. But, in practice, scientists require highly specialized vehicles to ensure a totally flawless delivery, making sure the precious cargo doesn't get destroyed or lost along the way. Let's look at Section 4, essential vector features. To pull this off, a vector absolutely must fulfill 4 non-negotiable features. First, it needs to be small itself, so it actually has room to integrate a large target insert. Second, it has to possess an origin of replication. We call this the ORI, so it can replicate autonomously once inside the host. Third, it needs unique restriction sites. If it has too many of these, the vector would literally just shatter into pieces when we try to open it. And fourth, it must contain at least one selectable marker. I want to expand on that 4th point really quickly because it's super fascinating. Selectable markers are essentially the factory's VIP bouncer or quality control mechanism. By using genes that provide resistance to specific antibiotics like tetracycline or ampicillin, scientists can easily screen out and destroy any host cells that fail to take in the delivery, meaning only the cells containing our vector survive the antibiotic screening. It's just a brilliantly elegant filtering system. And finally, Section 5, specialized vectors. First up in our fleet are plasmids. You can think of these as the standard reliable delivery vans of the microscopic world. They are circular, double-stranded, extrachromosomal DNAs that occur naturally in bacteria and even yeast. They can range from just a few thousand base pairs up to well over 100 kilobase pairs. And the best part? They are perfectly capable of replicating completely independently of the host's main chromosome. That guarantees at least one copy gets passed to each daughter cell during cell division. Next, we have phasmids, or phagemins. These are basically the amphibious vehicles of genetic cloning. They're true combination vectors. Their middle region is a linearized plasmid, but their ends are actually made of lambda-phage DNA. What does that mean for us? Well, it means they can be packaged in a lab just like a virus, but once they infect and get inside an E. coli cell, they switch gears and replicate just like a standard plasmid. It is truly the best of both worlds. Then there are expression vectors. Now, these are meant strictly for mass production. Rather than just dropping off the blueprint and leaving, they act as strict factory managers that literally force the host cell to efficiently manufacture the protein encoded by our clone gene. They achieve this crazy high level of control by using inducible promoters, unique insertion sites, and specialized transcription termination sequences. So this fleet of vehicles is already pretty impressive, right? But what if a specific logistics route requires crossing the boundary between totally different kingdoms of life? Like, what if we need a single vector to survive and replicate in both a prokaryotic bacterium and a eukaryotic yeast cell? Well, enter the shuttle vectors. These are the ultimate all-terrain vehicles in our DNA technology. They are highly versatile DNA constructs specifically designed using recombinant techniques so they can replicate in two completely alternative hosts. Because they're built with multiple origins of replication, one for bacteria and one for eukaryotes, they can seamlessly shuttle back and forth, operating in totally different species without missing a single beat. As the chapter so perfectly summarizes, quote, "Thus, gene cloning may be considered as a two-component system, a compatible host and a vector. The entire magic of biotechnology really does boil down to this perfect partnership. The vector provides the essential sequences required for replication, and the compatible host factory supplies the vital enzymes and proteins to actually do the heavy lifting." So as we wrap up this explainer, I want to leave you with something to chew on. If a single, beautifully engineered shuttle vector can effectively move DNA between a simple E. coli bacterium and a complex eukaryotic cell, what might be the ultimate limits of this microscopic manufacturing system? The possibilities for medicine, agriculture, and science as a whole are just staggering. Thanks for joining me on this deep dive into the microscopic logistics of life. Catch you next time.